Well, this is actually a very special moment for me because I am transitioning from one of my postdoc mentors to my current postdoc. So with that, talking about brain food, feeding omega fatty acids to chickens, we have Rosie Whittle. And somehow Darren figured out the pointer, so I don't know, be a wizard.
Hello, everyone. Thank you very much for having me here today. So a lot of what I'm going to talk about today is stuff that I did for my PhD project, and it's kind of come. How?
Loading. Yep.
Okay. After doing it as part of my PhD, it's kind of become a bit of a passion project for me. So if you ever want to talk about omega-3s, I'm kind of the person who will talk to you for hours about it, but I'm going to try and keep it to a relatively short talk today. It's going to work. Okay, so briefly what I'm going to go through, I'm going to give you a brief background on what omegas are because not all of us are nutritionists. I'm not a nutritionist, so I learned all of this whilst I was doing my PhD because I'm an ethologist from training. I'm going to talk about different sources, and most importantly, I'm going to talk about balancing omega-3s with another group of omegas called omega-6s. We're then going to talk about why omega-3s are fed to chickens, and also some benefits, talk about how they interact with the chicken brain and any behavioral effects of feeding omega-3s to chickens. So omega-3s are a group of polyunsaturated fatty acids. Okay. Is that any better?
Working, so you
Okay.
Speaking to here if you can. Or use one way.
Okay, is that better? Okay, so they're polyunsaturated fatty acids, which are categorized by long hydrocarbon chains. And specifically, they have a carboxylic group at one end and a methyl group at the terminal end. And this is why they're called omega-3s or omega-6s, because it's the last letter in the Greek alphabet. So the reason they're differentiated between 3s and 6s is basically the location of the first double bond from the methyl group. So omega-3s have the first double bond on the third carbon, whereas omega-6s have it on the sixth carbon. So I'm going to briefly go through the biosynthesis of omegas. Firstly, I just want to highlight it that omega-3 and 6 both have an essential fatty acid. So this basically means that it must be ingested from the diet because it can't be biosynthesized. So if you don't eat it, your body can't do anything to generate longer chain fatty acids. There are several very important omega-3s, one of which is the precursor, but also some longer chain fatty acids, which are essential for basically all biological functioning, one of which is eicosapentaenoic acid, or EPA, and the longest chain one, which I would argue is probably the most important, especially for brains, which is docosahexaenoic acid, or DHA. But not to forget the omega-6s, they are also very important for functioning and highlighting the precursor, which is linoleic acid, or LA, and arachidonic acid, which is AA. The most important thing about these two groups is that they compete for the exact same enzymes when they're elongating. So the same desaturators and elongases. So how much you eat is very important because they directly compete with each other. When it comes to different sources, I'm first going to go for omega-6s and highlighting some of the most common ones that we see. And some of the things that we feed to chickens are very, very high in these omega-6s. For example, in corn oil, sunflower oil, soybean oil. These are all very, very rich in omega-6s. And then when it comes to arachidonic acid, the other important omega-6, this is very common in all of these animal products that we commonly eat. So pork, chicken, eggs, salmon, they all have a relatively high content of arachidonic acid. Sources of omega-3 are a bit rarer, especially when we think of our current diet that we eat as humans. Oily fish, you've probably heard, is one of the best sources of omega-3s. And they predominantly have the longer chain omega-3s. They are very good at biosynthesizing and creating the longer chain fatty acids. When it comes to the precursor, this mainly or predominantly comes from plant sources, so grains. So we have chia seeds, we have flaxseed oil. Soybean oil also has a bit of omega-3s in, but it's not as common as, say, getting DHA or EPA from the oily fish, but it's still important. I'm going to emphasize these guys on a sort of a nutritional scale, that it's important that we're having an equal quantity of both in the diet because of the biosynthesis. So we want to eat an equal amount of omega-3s and 6s so that, say, hypothetically, we have 6 available enzymes and we eat 12 molecules of 6 or 3, we want them to have half and half equal representation. However, say we're having a very, very rich omega-6 diet, we love our grains, like we have cereal, we're having our bread, everything, and then we decide to take a fish oil pill every now and again. So we're having a very small intake of omega-3s. This is creating some imbalance in our diet, meaning that there is no longer this equal synthesis or biosynthesis of these omegas. So from now on, I'm going to try and talk of omega-3 and 6s on a ratio scale. So I'm going to talk about the omega-6 ratio. So we know that corn has one of the highest grain omega-6 to 3 ratios that you can get. It's a 50-to-1. Soybean is a lot lower. It's at about a 7-to-1. And these are the most commonly used grains when it comes to chicken nutrition. We have some things in sort of the central, more equal 1-to-1 ratio of omega-6s to 3s. In terms of chickens, we sometimes feed like spirulina to chickens that has a 1-to-1 ratio, or chlorella is another seaweed species which has a 1-to-4 ratio. And then we have our fish oil or oily fish, which has a higher omega-3 content, and also then flax seeds, which have a higher omega-3 content. So when it comes to commercial chicken diets, we want to try and keep the cost of these diets down. We want to use a high yielding grain that we can produce in bulk, that's not going to cost us a lot to produce. And then on top of that, we're not just including the grains into the diet, but we often use some kind of an oil to help bind the diet as well. And these oil supplements are also a corn or a soybean derivative. Now, the diets of these the diets may differ in omega-6 to 3 content depending on the region because of the grains that they use. For example, in Europe, they use a lot less of the corn and tend to use more of a wheat-based diet, just because that's what the climate allows them to grow better. And wheat has a lower 6 to 3 ratio than corn does. And you can see this reflected in the omega-3 to 6 ratio content of table eggs, for example. In the US, the average omega-6 to 3 ratio of a table egg was 19-to-1, compared to 9-to-1 in European table eggs. Now, feeding omega-3s to chickens isn't new. This has been done since the 80s. I think the first research into this was done by Steve Leeson and Linda Caston, where they were feeding flaxseed to hens to enrich the eggs with omega-3s for human consumption. More recently, people have also been exploring feeding omega-3s to broilers to produce omega-3 enriched broiler meat. And again, they predominantly are feeding flaxseed. Now, in the past, they have experimented using fish oil, but it does affect the sensory characteristics of the meat and eggs. Nobody wants to eat chicken that tastes like fish, for example. And the reason why they're doing this is because they found that chickens are actually relatively efficient at biosynthesizing ALA to DHA in comparison to humans. Now, I haven't found any sources which have backed this up. This is just what I've been told, that they are more efficient than humans. When it comes to humans, we convert ALA to EPA at about 8%. When it comes to ALA to DHA, we're rubbish. We can't do it very well. Some people have a 0 conversion from ALA to long chain DHA. Other people have up to a 4% conversion. So pretty inefficient. So if we can find ways to get more omega-3s into the diet by using chicken as a conduit, that is great. But my biggest question is, how can this affect the chickens? If we know omega-3s are good for humans, can they also benefit the chickens themselves? So a lot of this research, which I was looking into, looks at sort of fetal programming. So this is looking at maternal nutrition in order to get the right deposition of nutrients into the egg to support the embryo in development so they can develop as completely as we want them to. Some current research into this has found both health benefits and also musculoskeletal benefits of maternal fed omega-3s. We found that it could increase the cortical bone development of laying hens, and also when fed to producing hens, it has been shown to decrease keel bone damage severity. In terms of immunity and health, it increases the antibody responses to a challenge and also has shown to decrease cardiovascular morbidity in broilers. So this is going to lead me on to the brain stuff, and everything we've heard today has been amazing about the brain and about the visual system, but I would argue that omega-3s are the building blocks of all of those things. So we know that the brain is very, very rich in DHA, so the longest chain omega-3, and it's very, very important for brain development. So for the chicken, this is embryonic brain development in the egg. And we can show that omega-3s preferentially accumulate within the brain during these periods of brain development. We know that it's very important for the structure and function of lipid rafts and neuronal membranes within the brain. Omega-3s act as direct ligands to transcription factors for genes, and these are genes relating to neurogenesis, so the creation of new neurons, synaptogenesis, the creation of new synapses, inflammatory responses, cell differentiation. Omega-3s are also precursors to the biosynthesis of different lipid mediators, specifically those relating to immune response. For example, omega-3s are the precursor to anti-inflammatory cytokines. Some literature in mammals, looking at a larger perspective of omega-3s in the brain, has shown that just maternal feeding has been shown to increase the volume of the brain, the gray matter, and the hippocampus. I'm highlighting the hippocampus specifically because as a behavior person, we know that the hippocampus is important for learning, as well as episodic and contextual memory. So how a chicken is able to learn about and perceive their environment and then use that information for the future. One of the biggest questions is, when is supplementing the most important part? And what is the best time point for supplementation? And this really depends on the species you're doing. So I'm going to quickly segment into precocial versus altricial species. We know that chickens are precocial. This means is when they hatch, they have a relatively developed functioning. They can get up and run, they can see, they can feed themselves, which is good for us because it means that they don't need to be raised with a mother hen. They can find food on themselves and be raised pretty easily. On the other hand, you have altricial species, which are born pretty helpless. They usually cannot thermoregulate. They don't have fur or feathers. And they present a dichotomy in terms of brain development and accumulation of omega-3s within the brain. So for a precocial species that needs to be up and going from the moment they hatch, we see that in the brain, they accumulate DHA. The peak is before hatching. And if we compare this to an altricial species, this occurs after hatching. And that's the graph on your left-hand side. And then on the right-hand side, we see also a dichotomy in terms of just general muscle accumulation of DHA. So this is showing that before hatching, chickens accumulate most in their muscles before hatching, They're basically setting themselves up to be able to get up and run as soon as they hatch. Whereas for the altricial species, this all occurs after hatching because they sit in their nest. They're protected by their parents. They don't have to get up and run until they're ready to fledge at whatever age that may be. So when a chicken ingests these omega-3s, she accumulates in their body, in their brain, in their muscles, in their organs, and then also can deposit these omega-3s into the egg for the offspring. These developing embryos that utilize these omega-3s during development. So I just wanted to give a quick background on my PhD. This work was all conducted at the University of Guelph in Canada, and it was part of an extensive project looking at the nutritional approaches for looking at productivity and welfare in poultry using maternal feeding of omega-3s as the conduit. So this was conducted by 3 PhD students, 2 nutrition students, and then myself. They each took half each of one did broiler breeder work, one did layer breeder work. I had the honor of doing both, so I got to experience everything during my time there. And the specific objective was me just to look at the behavior of these chickens and how maternal feeding affected their behavior. So I had 2 different maternal diets. One was a standard chicken ration that was formulated. And then we had a diet which used flaxseed. And this is the source of the precursor omega-3 ALA. We fed the diets in a 2x2 factorial design. So we had a rearing diet where half of the broilers were fed the control or flaxseed diet. And then at 19 weeks of age, we switched half of them to the opposing diet to create a rearing by laying diet combination. It's important to note that these diets, we did try to make them as equal as possible. So they were isocaloric and isonitrogenous. We balanced the metabolizable energy and crude fat, protein, and fiber as much as possible. And they really only differed in the omega-3 to 6 content. So for the control diet, they had a 6:3 ratio of 26:1, whereas for the omega-3 diet, they had a ratio of 4:1. So that's really where they differed. And then we hatched 2 cohorts of offspring. I did a similar experiment with laying hens. We used 2 different strains, a brown and a white feathered strain, again, feeding the same inclusion of flax seeds, balancing the diets for metabolizable energy, fat, protein, and fiber. And again, they only really differed in the omega-6 to 3 ratio of the diets. And again, 2 cohorts of offspring. So one of the first things we wanted to do was to prove the concept that, yes, we know that breeders can deposit omega-3s into the eggs, but can we say that, yes, the offspring uptake these omega-3s? So what we did is we conducted brain dissections at 1 day of age. So we took chicks straight out of the hatcher and sacrificed them for brain dissections. And we firstly calculated the brain to body weight ratio of these chicks. We then went on to analyze the brain fatty acids to see, well, have we influenced them in any way? So this is the first result I want to show you. All of these graphs are laid out the same way, with broilers are always on the left and the layers are always on the right. The control diets are always in the dark blue and the flaxseed diets are always in light blue. So on the left with the broilers, we have just the laying diet and the brain to body weight ratio on the y-axis. So we found that in broilers, our maternal flaxseed diet increased the brain to body weight ratio of these broilers. However, when we went to look at the brains of the laying hen offspring, we didn't find any effect of maternal diet on brain size. When it came to the brain omega-6 to 3 ratios, for the broilers, we didn't find any effect of maternal diet on the brain fatty acid contents, which was surprising for us. But when we looked at the laying hen diets, we found that the control hens had a higher omega-6 to 3 ratio in their brain than the flaxseed hens. In fact, the flaxseed hens had a 1-to-1 ratio, which is, some of the literature says that this is the gold standard for ratio that they should have. One other paper has ever looked at this before, so that's my comparative literature section for this research. A study conducted in 2003 fed differing maternal diets. One was a sunflower oil-based diet, one had an equal quantity of sunflower and fish oil, and then one was just a fish oil-rich diet. And they found that these diets successfully altered the brain omega-6 to 3 ratio. And this was done in broilers. Compared to what I found in my PhD, we didn't find a difference in the ratios, but we found brain size differences. And in the layers, we found that it affected. So their results matched more our layer results than it did our broiler results. And this is an N of 2 for studies, so please more people do this because this is all contradictory, so we need to confirm some of these findings. You might ask, well, why do we care? Why do we care if maternal omega-3s affect chicken brains? Well, a lot of literature, this is kind of a spoiler alert for what I'm going to show later. A lot of comparative literature shows that omega-3s can increase fearfulness of offspring. Not so good. But also, it has been shown to increase cognitive ability. So I'm going to go through those two factors now and explain some of those results. So some of the implications of fearfulness in poultry is that it can be very problematic in commercial systems, especially when animals are experiencing chronic stress. In terms of fear itself, this can pose a big welfare issue because it could increase the risk of injuries, especially in relation to like panic smothering events. We talked about smothering briefly. There's kind of two types. One is a panic smother, and one of them is a creeping smother. So we really don't want to have hens with an exaggerated fear response because we don't want to have any mass mortality risks in our flocks. So one of the tests I conducted in my PhD was called a social isolation test. Now this test was validated pharmacologically by Kenneth Sufka, and the whole test they conducted lasted 60 minutes, and they called it the anxiety depression model. But I'm going to just focus on the first 5 minutes of this test, which is the anxiety phase. So they characterized the anxiety phase as a period of high levels of vocalizations, and then after 5 minutes, this decreases into what they call the depressed phase of this test. So I conducted this test for the first 5 minutes looking at the anxiety. And we recorded vocalizations as well as any freezing behavior and escape attempts. Today, I'm just going to present the vocalizations because I could speak about this for hours. So again, we have the broilers on the left, just focusing on the laying diet of the broiler breeders and the number of vocalizations. On this graph, we have males and females. So we have females as the solid bars and males are the pattern bars. We found that flaxseed males vocalized significantly more in this test than all of the other sex by laying diet combinations. So this indicates that these flaxseed males or males from flaxseed fed mothers showed more anxiety or fear during this social isolation period. When it came to the laying hens on the right, we didn't find any effect of maternal diet on their fear behavior. In terms of other literature has also shown increases of fear due to maternal fed omega-3s. This study looked at feeding fish oil to the parents. So that's the MH chicks the parents were fed fish oil. And one of the tests they did was looking at the reaction to novel food. So they call it familiar food and unfamiliar food, but they were both cracked corn and they were both novel food. However, one of them they added fish oil to, so it had similar sensory characteristics that the parents' diet had, not their own diet. And they found that with these MH chicks, the fish oil chicks, they had a shorter time spent eating the novel food than all other treatments. This indicates that they were more cautious of it. They're perhaps more fearful of the novel food. They also conducted tonic immobility. This is the graph on the right. And they showed that the fish oil chicks had a longer tonic immobility, indicating higher levels of fearfulness than the control chicks did. Interestingly, they also did tonic immobility in the parents and found no differences of diet. So it was only really the offspring that were affected by the maternal diet. Another study looked at the reaction to a novel object, again using this fish oil compared to a control diet. And they found that the chicks from parents fed fish oil had a longer latency to eat from a novel feeder and spent less time eating, indicating that they had more fear of the novel object or feeder. What about other species? Because that is the chicken literature. There is no more. It's 3 studies. So comparative literature it is. In mice, it's a bit contradictory. It's shown that have increased fear of novel objects, increased anxiety responses in plus mazes, but only under stressful conditions. So under non-stress conditions, there was no effect of maternal diet on anxiety responses, but when we stress the mice under very high light intensities, this is when the behavioral difference showed between the 2 treatments. Another study showed that it increased locomotion in an open field. If you're not familiar with an open field test, high locomotion means low fearfulness. So this is where the contradictory research comes in. In Muscovy ducks, this is also contradicts some of the literature in chickens because they found maternal fed omega-3s decreased offspring tonic mobility, so they were less fearful, but they didn't find any effects on social isolation stress. I just want to throw humans in there because most of the literature in omega-3s is human biomedical research. And they have found that when using omega-3s as a nutraceutical, so this is feeding omega-3s in conjunction with something like an anxiolytic, they found that people had a reduced level of anxiety. However, none of them fed omega-3s alone. They've always paired it with an anxiolytic, so there's a bit of a gap in that literature. So now I'm going to talk a little bit about cognition. So why is cognition important? Do we want chickens to be smart? To some extent, I say yes. Because chickens are precocial. I've established this in this talk already. They're raised in an age homogeneous environment. So they learn from each other or from us briefly showing them where food and water is. But in a commercial barn, if you show 10 chicks, that's maybe 1% of the flock, 0.01% of the flock, depending on how big your flock size is. So they learn from each other, they learn from experimenting, and they have to have a certain cognitive ability to be able to do that for themselves. They need to learn the relocation of resources within their environment. In laying hens, we're putting them into aviaries now. They need to be able to navigate these complex housing systems. They need to not just be able to see things, they also need to be able to learn the best pathways to get somewhere. They need to be able to identify what their abilities are in these systems. So one of the tests I did to test cognition in chickens is I conducted a 1-trial T maze test. So this was a maze shaped like a T, and we colored each of the arms differently, 1 green and 1 blue, just as a visual cue. And then we placed a mirror in one of the arms as a social reward. We know chickens are social. So if they see the mirror, they don't see themselves, they see another chick. So it's a social reward for them. I'm hoping this video will work. Yes. So to do this test, we had a habituation period. So 3 times 1 minute. And we had 3 mirrors in the maze at this point, just so they could experience the maze. So they had some free exploration time before we then conducted a bit of training. In the training, we just had 1 mirror in. So here it's in the blue arm where the right line is, the red line is. And we just pushed them gently into the arm and then into the incorrect arm so they could experience both experiences. So during testing, those yellow lines there were participation criteria. So they had to cross the line to be able to have participated in the test. And if they didn't make a choice, after 60 seconds, they were removed and replaced. So the mirror here is in the green. This chick makes an incorrect choice. And this one makes a correct choice in the green arm. So what did we find? Well, I'm going to preface this as we didn't find anything in terms of the number of successful choices they made, but we did find something to do with the number of choices they made. So we combined the incorrect and correct choices as they were participating. And we found in broilers, the maternal diet affected the number of times that they actually made a choice at all. It doesn't mean that they were smarter or not, that they just participated in the test. So again, we found this was a sex-specific effect. And this time we found that females from flaxseed-fed mothers were more likely to participate than the other treatments. I'm just going to note that this was a difference of one choice. So it's not a huge difference, but statistically it did show that there was an increase in participation. And in the layers, again, you've probably guessed it, we didn't find any differences in this test. I couldn't find any other cognition literature in chickens to do with cognitive ability and omega-3s, but there is some comparative literature. One study in partridges looked at passive avoidance learning. So this is basically presenting day-old chicks with a bead, and then because they have this natural instinct to peck at everything because they want to know what they can eat and what they can't. So they presented a red bead and a blue bead, and then they dipped the red bead in a bitter taste and wanted to look, would they remember that this red bead tasted bad? And they found that yes, maternal fed omega-3s increased their ability to remember that they'd eaten something bad previously. In dogs, there's been research that's shown that feeding puppies omega-3s increased their object discrimination ability by about 10%. There's an extensive literature in rodents because of the biomedical comparative human side of biomedical research that shows that maternal fed omega-3s increase spatial and working memory. And even in humans, if you're having kids, they always tell you to load up on omega-3s because it's important for the baby. Well, yes, eating fish 4 times a week has been shown to increase development scores when it comes to social skills and communication in children. So just to quickly summarize everything I've thrown at you, we found differences in brain size and omega-6 to 3 ratio in broilers. We found that it can affect behavior in some aspects, but not all. The differences were very sex-specific and not very well defined. In terms of the layer research, this is mainly on me. We found an altered brain omega-6 to 3 ratio. I didn't find any cognitive effects, but I'm an N of 1, so we need more research to explore this. In terms of fearfulness, we find that some research shows that maternal fed omega-3s can increase fearfulness in laying hens. I'm going to quickly touch upon why I didn't find results in the layers by going back to the diets. So my control diets had a high omega-6 to 3 ratio, which resulted in a high ratio in the eggs compared to the flaxseed diet. But when I looked at the same for the layers, the control diet already didn't have as high ratio of omega-6s, but they appeared to be damn good at converting these low numbers of omega-3s in their diets to producing more in the eggs. So they had a low omega-3 ratio in the eggs despite not being fed that many. In fact, it didn't really differ from my flaxseed treatment at all. So I'd say the main issue is that the control diet was not an effective control for this experiment. The key difference between the diets is the broiler breeders, we fed a corn oil supplement, whereas for the layer breeders, it was a soybean oil supplement. So if you remember, corn oil was 50 to 1, soybean oil was 7 to 1. So that was where the difference in the ratio really was for these diets. I'm now going to move into my next little pet project is, well, what about wild diets? What what would a chicken normally eat in their diet. So some comparative literature for pheasants, partridges, and ducks always show that the captive diet has a much higher omega-6 to 3 ratio than the wild diet does. In fact, the wild diets are almost as close as 1-to-1 on average. But what is the right ratio for chickens in terms of their development? Well, in my study, we showed that just by adding flaxseed, we can get pretty low ratios of omega-3s to 6s. There's some research conducted in the 80s on Greek islands where they looked at free-ranging chickens, and they found that just letting them free range in olive groves, they could achieve an omega-3 to 6 ratio of 1.3 to 1. So that's pretty close to unity. There's even one company in Belgium which is coining a wild type egg. So they are specifically feeding them a diet to achieve what they call as a wild type egg, which is a 1-to-1 ratio. And they're doing this for human benefits, not for the chickens, I might add. So we don't actually know what this ideal ratio for chickens is. So I just wanted to highlight a bit about what they would come across in their natural diets if they were in the wild. Well, one of the first studies that looked at natural diets for domestic chickens in the wild looked at putting chickens on Scottish islands and letting them go and see what they did. Now, this was conducted by Savory, Woodgush, and Duncan, and they literally just decided to up some chickens and put them on a Scottish island and look at everything. So they observed that in the spring, they ate predominantly grass, whereas in the autumn or fall, they ate predominantly wild oats. So already those have a pretty low omega-6 to 3 ratio. One study in Bermuda looking at feral chickens. So these were domestic chickens, which then, due to adverse weather conditions, got free. And they were found to eat a wide range of diet, including different vegetation, insects, as well as human garbage and commercial garbage. And the study which I talked about on the Greek islands, they highlighted what was probably causing this very low omega-6 to 3 ratio, which was a plant called purslane, which has a 1 to 3 ratio, very high in omega-3s. And they said that this was most likely the reason why they were getting an egg close to 1-to-1 ratio. And this is where I would like to liken chickens to dinosaurs, because they are not vegetation eaters. They're not granivorous. They don't eat just grains. They'll eat small rodents. If you ever seen a chicken eat a mouse, it's kind of graphic. They can eat lizards, playing snakes. So they do eat a very, very varied omnivorous diet, which is different to what we are currently feeding them in commercial housing systems. So what I really want to take away from this talk is that commercial diets for breeders may not provide the ideal ratio of omega-6s to 3s in the brain to facilitate offspring brain development. I mean, yes, they grow really well, they're very productive, but in terms of behavioral aspects, maybe it's not what they would experience if they had a natural diet. But we don't actually know what this correct ratio is yet. I think it's very important to weigh the cost and benefits of increasing cognition versus increasing fearfulness. Because I think every producer out there would be like, we don't want scared chickens. We don't want them to be fearful. Maybe it's better for them to be a dumber chicken and less fearful than to be a more fearful chicken and more cognitively able. One thing I haven't talked about today because I didn't have time, but I'd love to discuss with people is the potential for mediating abnormal behavior. There's extensive comparative literature which looks at omega-3s and abnormal behaviors in humans, in mammals, in other animals, and it's shown to reduce abnormal behaviors. So that is a very interesting aspect which hasn't really been explored yet, which I think has a good potential for the poultry industry to explore in the future, which may outweigh the negative fearfulness costs if we can reduce abnormal behaviors. That is one option. So I'm going to finish it there. I just wanted to acknowledge my PhD committee at Guelph because they really made this work happen, and all the funding bodies in Canada which allowed me to do this research. I'd like to also thank my current supervisor, Dr. Weimer, firstly for giving me the postdoc position here in Arkansas and allowing me to speak to you today about my little passion project, and also the Weimer Lab and everybody at the U of A for supporting me in my move. So thank you very much.
We have a good amount of time for some questions.
Oh, that's fast.
First, Dr. Kidd, no nutrition questions? Or are you just amino acids? I'm coming here anyway. Better think of one, Mike.
Hi, can you hear me? Yes. In the studies where you show different other species that receive the omega-3 diet, were the diet administered only during pregnancy, or was the diet also administered after to the offspring?
So I kind of chose studies which highlighted that developmental period. So some of them were maternal feeding and some of them were during lactation. So it really depended on the species. So for the bird studies, it was all maternal feeding. But like for some of the rodent and so the dog stuff, it was more like offspring feeding, but also during lactation, because that's when their brain development is at its peak.
So during lactation, were the mothers still fed with the omega-3 diet?
Yeah, so the mothers were fed the diet and then it transferred to the offspring through the milk.
Until weaning, I guess, when the offspring are separated from the mother cage.
So I didn't really include that much. I think there was the one dog study, which was on 3-month-olds, and they just gave them a fish oil pill. So it does really vary. There's not a lot of literature out there, so I really just had to take what I can. And I pretty much presented all of, apart from the mice and rat stuff, the other species, that is all that there is out there. There's very, very little literature looking at omega-3s and behavior, which is kind of unfortunate because there's so much in the mice and rat and human literature that all of the animals it's an open field for everyone to go and explore.
Thank you.
Thank you. It brings me back to my extension days when I had someone from the public ask me why their mother hen wasn't lactating. Any other questions over here? All right. Okay. I'll ask mine on the way. So I think a common theme that we've heard today is the differences between males and females and a lot of the experiments and results that were presented. Do you think that your male results were because of the mother hens that were fed the flax or just the sex differences?
I would like to say it was just the maternal diet because we didn't see increases in vocalizations from males in the control groups, for example, but there does seem to be some kind of interactive effect between sex and maternal diet. Because if it was just maternal diet, then we'd expect males and females to both show more vocalizations. But we just saw it in that one group. So it was different from females of their own, like their own sisters. And it was also different from those from control fed males. So it's one study. Based on that one study, yes. But I would love to explore it more to try and increase the body of literature because we know that one study does not prove concept of anything. We really need to get down and see exactly what's going on.
Definitely, but I also think that we're seeing there's some similarity maybe with Victor's project with the cognitive judgment bias where we saw that interactive effect of handling, I think. Very, very interesting.
We have a couple questions from our virtual attendees. From Sarah Ballard. For the flaxseed diets, does it manage hormones in order to calm anxiety in humans? And why does this differ in chickens?
Does it manage hormones? I'm not sure, to be quite honest. All I know is that We fed ALA, and chickens can successfully convert this into longer chain fatty acids that are very important for things within the brain, which can include mediating the serotonergic, dopaminergic systems. Whether we can say that this is causation or not, I don't think we can, but there is pathway links for them. Just nobody has explored that. So it's only going step by step that we can actually show that kind of concept, but nobody has done it yet.
Thank you. I have a small question, but I don't know if related with the talk about behavior. I don't know if you have the chance to see the liver or if there is a reducing the fatty liver using omega-3 synthets, or how you see the liver score in this verse, or you don't make.
I didn't look at that myself, and I'm not sure whether any of my colleagues did as well, to be honest. It is an interesting concept because we're feeding fat, but it's not. I think the polyunsaturated fatty acids are not the biggest cause of fatty liver disease. And it's merely the ratio of the two that we're manipulating. So I can't really speak for this because we didn't look at this, and it's not my field of expertise, but I haven't seen anything in the literature to correlate those two things yet.
Hi, great talk. I have a question. You measured the relative brain size. Can you talk a bit about like the direct link between brain size and cognitive ability in the birds?
Yes. So it's a bit of a tentative link. I mean, it's just generally assumed that brain size equals greater cognitive ability. On a species level, I'm not sure how accurate this is. We like within all chickens, we don't know that if one chicken has a bigger brain, that it's more smart. But we know from comparative literature in birds in general that like corvids and parrots, they have large brain to body weight ratios compared to some other birds. So in terms of all birds, yes, you can link brain size and cognitive ability, but I'm not sure on a species level how relevant it is. It's tenuous at best, I would say.
I guess.
Hi, really cool. Just to mention that last question, just a point, it's not brain size to body size. It's not terribly meaningful cross species. In mammals, it's more about cortical thickness relative to volume of brain, is related to cognitive ability. But bird telencephalon's cortex is organized completely differently, and that would be difficult to do. Coternyx, the crows and ravens, are typically considered some of the smartest birds, and they have relatively small brains. Anyway, I'm more interested in that fearfulness, that increased fearfulness of the offspring. I mean, that keeps coming up. And when you look at the wild bird literature, that they are very much believe that increased fearfulness is a good thing in the offspring in preparing them for harsher climates, environments, or whatever, because they will take fewer risks. They will have a more rapid, stronger immune response, with, like, for example, the HLR, and glucocorticoid response, and also take fewer risks. So, I mean, what do you think? I mean, how does that relate to birds in a cage-free house or caged houses or whatever?
In an ideal world where all chickens live free, yes, being smart and fearful is good. But in commercial housing systems, I don't think we want this, purely because of the systems that we manage them in. Because if we have smart but fearful birds, it can compromise their welfare in terms of just generally fear is negative. We know that if they have chronic fear, they tend to be in a negative affective state, so which is bad for their mental well-being. We know that it could increase events